I'm Steve Cook, and I'm going to present to you this work on genomics pipelines and how we're going to use these to hopefully provide a lot more genetic data in the hands of medical professionals who need them. This is work with my mentors, Jihoon Kim and Dr. Claudio Farkas. And so let's start by way of introduction. Why do we care about genomics? What we see here is a slide from the 23andMe project, provided you registered before they ran afoul of the FDA. So the information on the left side, the disease risks, they're not allowed to report anymore. But this is an example of the kind of data we can glean, these very important statistics that we want to be able to get fast and affordable access to for clinicians in a medical setting. So things like drug response, carrier status, and risk can be very useful in evaluating patients, what difficulties they might be having. So for a very short crash course in genomics here. Inside the cell, there are 23 chromosomes. Each of these chromosomes is composed of tightly wound DNA, which is itself composed of the four molecular letters, A, T, C, and G. There are about 3 billion of these base pairs if you lay all the chromosomes out from one end to the other. Now, a small fraction of these, 1 to 2%, actually code for proteins. They're transcribed within our cells into the proteins that give us our function. This subset is called the exome. And so it's a much easier computationally to examine the exome and still get lots of very important information from this. So that's what we're going to be looking at today, is exome sequencing. So how do we figure out what someone's exome is, and how do we get it into a digital format where we can run a computational analysis on it? So current next-generation sequencing machines will fragment someone's DNA, replicate it many times over, and then attach adapters, scan it, and provide a 100-gigabyte file or more of all of the DNA information in a file format. So now it's up to us to then take this and figure out how to take all these little pieces and put them together. Each piece is only on the order of about 100 base pairs long, and we're trying to line this against a 3 billion pair reference. So looking at this in a little more detail, we see these small sequences here, and we're trying to line them against the reference genome down at the bottom. So it's a very big jigsaw puzzle we have to do. But it's not that easy. Not everything is going to match perfectly. There are errors in the sequencing equipment. The person's data might be very different from the reference we've collected. They might be missing things. They have extra things replicated. So it's a very difficult problem. It takes complicated statistical approaches in order to deduce what the actual, in order to do this assembly correctly. So once we have this assembled, and we don't even know if our assembly is right, now we're looking for variants. On the left, you'll see a homozygous variant where every single marker for that individual is a flip, a SNP, a single nucleotide polymorphism at that position. On the right, sometimes they have it, sometimes they don't. And this can be very complicated. It can be due to sequencing errors. It can be noise. And we need to say, okay, which of these are going to be known markers for dangerous diseases and medical conditions? Which of these are noise? Which of these are benign? And that's the challenge we're looking at here. So we want to create a software pipeline that can do all of this work to go from those fragments that the sequencing machine gave us to something like the 23andMe output slide I showed earlier, with very little need for advanced computer science techniques, software configuration. We want to make this something that the person can use. So we want this to be fast. So we want to be able to do a human exome on the order of several hours so we can get the information as quickly as possible in a life-or-death medical situation. We want it to be accurate. We don't want too many false positives or false negatives when we're predicting what they might be at risk for. We want this to be affordable. We want to do the data processing for one person for the order of $10. And finally, as I was mentioning, we want it to be easy. It should be something that people who don't have strong computer science backgrounds can install and run. So we set out to evaluate some of the current systems out there and try to make them work for us. And we learned some things along the way. First lesson is that the existing software pipelines are, generously speaking, difficult. What you see here is an email chain started on July 8th with one of the developers of a popular pipeline. And it took 62 messages back and forth since then to get this thing configured and set up. Along the way, we found bugs in the code, parser difficulties, software dependencies, all manner of things made this as difficult as absolutely possible. I was shocked. and we're still learning more. The second lesson is that ground truth is still kind of the Wild West. What you see here is a many-way Venn diagram between several tools for doing the variant calling. All of them, you'll notice, report variants that the other ones don't. So every one uses a different underlying statistical model to do the variant detection with varying results. These are very recent papers. SNVer, I think, was 2 years ago. So this is all still area under development. So we think that a good pipeline should be able to support not just one, but many of these, interoperably so that the user has a choice of which one they want to go with if they decide that, say, GATK provides the best benefit for what exactly they're looking for. So after all this trouble, we've got a pipeline working, basic one, and we compared its chromosome. We looked at the chromosome 1 at the number of SNPs discovered. It passed most of them, and what you see in the middle column is how many of these were in a reference database that had been seen before. And we saw that for what we discovered, roughly 10% were novel for this individual that we tested. We compared our pipeline against what I call a SAM tools pipeline, doing many of these steps manually with command line tools. And we found a good deal of overlap in the number of SNPs discovered. Again, as we saw earlier, it's not uncommon for there to be a great deal of differences, but this at least gives us some confidence that our pipeline is similar to other existing tools out there. So we've looked at accuracy a little bit rather than glance at the surface. Now let's look at the single exome time and cost. So our pipeline takes 5 hours and 20 minutes to run on a powerful server here at UCSD. A published one takes 10 hours, and extrapolating these costs to a similar power Amazon machines to be rented from the cloud, we see that our cost is roughly the same, on the order of $8 for just the CPU processing time alone. I did some extra calculations for the S3 storage cost and the EBS, the volume store cost on Amazon, which is all the space you need to actually store these hundreds of gigs of data. And I came up with a rough number of about $20 per exome. So we're pretty good on our target of money here too. So to summarize the project and look forward to the future. So this summer, I configured a working pipeline for variant calling. It was a very difficult, very manual process. I have a CS background and 2 months at my disposal to just focus on this project, and it was still extremely challenging. I don't see how anyone who's a geneticist with very limited time and resources would be able to do this. Even published results did not work as intended. Another one, you'd kick it off on Amazon, and the idea is that you would give it your credit card information, step one, and your genetic data, and it would rent servers as needed to process the data and give you a result. After 4 days, it was still charging us money, still renting servers, but it didn't actually provide any data or output. In fact, it seemed to hang. So they were mining Bitcoins for all we know. So anyway, these published works do not work very well, and there's a lot of room for improvement for these tools that are very important. Some more flexibility is needed as well. We want something that can be configured easily and also run multiple different variant calling tools under the hood, since there's so many approaches out there and no one seems to be sure quite exactly which one is best right now. We also want to deploy for different use cases. We want an outcall script for organizations such as the VA Medical Center that are not allowed to send their patient data outside of their own system. They can't upload to Amazon. So we want to give them something where they can download and run this on their own servers locally, and then they won't have that problem. Also, we want to provide an Amazon Cloud Compute image. So let's say there's a small lab that cannot afford its own hardware. We want them to be able to use this Amazon system, and then on the order of, say, $20 like we'd seen, they can start getting into exome sequencing. Finally, contribute back to the community. With all the bug fixes I found, I got a lot of thanks and everything from some of these developers, and we think that making these tools better will eventually, in the end, benefit everyone. So finally, in conclusion, I'd like to say that we found lots of room for improvement, and we currently have our own tool under development that we think will solve these problems and provide a great benefit to the community, especially in light of the ever-increasing number of genomes and the terabytes upon terabytes of sequence data being generated. With that, I'd like to thank my mentors and the DBMI staff, interns, and program, and open up for questions.
Let me ask your personal opinion about 23andMe. How far is it from providing information that would be?
Well, with them currently, it's a full. They do the sequencing and everything. So we're envisioning more like a medical center that has its own sequencing department, and they can actually do the wet work themselves. It's the DNA processing after that was more what we were looking at in this particular instance. Part of the reason there is the controversy around them was because they were trying to provide this medical data without really being upfront about just how wishy-washy some of this is and the statistical confidence intervals and how much of this is still unknown. So I haven't tried it myself, but my understanding is that they don't report very much on what algorithms using under the hood. There's a kind of a lack of transparency, I believe. And we want a tool that will provide every single step of the way it can be configured, and you'll know exactly what's happening end-to-end. Also, faster, more high-performance setting. If you're in a clinical setting, you don't necessarily have the week or whatever it takes to do the spit swab, mail it to them, and mail you back. Thank you.